Hello friends, today we will going to discuss about Staphylococcus aureus microbiology question. So what are the objective of discussion? Morphology, cultural characteristics, pathogenicity. So what is the morphology? Streptococcus or Staphylococcus aureus are spherical cocci approximately 1 micrometer diameter arranged characteristically in a grape-like cluster. Cluster found each single or in pair or chains of 3 to 4 cells. Streptococcus or Staphylococcus aureus are non-motile and non-sporing. Non-capsulated strain having some amount of capsular material on the surface which stain readily with aniline dye and are uniformly gram-positive. On the influence of a penicillin dye, they may change into spherical to L form. Now we go towards the cultural characteristics of a Staphylococcus. Staphylococcus grows readily on ordinary media within a temperature range 10 to 42 degree in pH 7.4 to 7.6. Staphylococcus are aerobes and facultative anaerobes. In nutrient agar, after 24 hours of incubation, the colonies of Staphylococcus are large, circular, convex, smooth, shiny, opaque and easily emulsifiable. Staphylococcus aureus show a golden yellow pigment on most of the stain. So the color of a pigment which is show in any type of a stain is golden yellow. The pigment produced occur optimally at 22 degrees centigrade in the aerobic culture. Pigment production is enhanced by giving glycerol monoacetate in the 1% solution. Then in nutrient agar slope we see the Staphylococcus shows a confluent growth, means which having a characteristics oil paint appearance. Same result also find in the blood agar, so we called as blood agar is similar to that of a nutrient agar. Now, the most of the stain, that is Staphylococcus stains, are hemolytic, means they especially incubated at 20 to 25 degree centigrade in the CO2. Staphylococcus aureus also grow in MAC conche medium, produce a smaller colonies which are in pink color. So in MAC colonies, MAC medium it produce pink color, whereas in medium it is so golden yellow color. Okay, then Staphylococcus also grow in MAC conche we discussed. Then some selective media also used for isolating Staphylococcus from specimen such as fecal containing other bacteria. So there is some selecting media used. The media contain mainly 80 to 10% of sodium chloride, lithium chloride, telluride and polymyelin. So human blood not used for the media purpose as it contains the antibodies. So antibody antigen deglutination occur, so that the stain is not isolated. That's why human blood agar is not taken inside for the staining. Now the pathogens, that is infection and intoxication. In infection there is damaging to skin, muscle, mucous membrane, drug adhering to of the cocci to the extracellular matrix. Then Staphylococcus invade the defense mechanism and multiply and causes tissue damage. How the Staphylococcus enter to the body? First it invade the defense mechanism, then enter into the body and multiply its number. In intoxication disease, it is caused by the bacterial toxin which is produced by either in the infected host or performed toxin in vitro. So the virulence factor present inside the organism which is the which produce the clinical syndrome. So in this way they produce the pathogen, either infection or intoxication. So intoxication is due to the bacterial toxin. Okay, now the virulence factor are also present which causes the clinical syndrome. Now what are the cells associated polymer present inside the Staphylococcus aureus? They are cell associated polymer, means they contain the polymer mainly the cellular polysaccharide or peptidoglycan confers the rigidity and the structural integrity to the bacterial cell. This mainly causes the activation of a complement syndrome and release the cytokinin. And also there is a telluric acid also present in the cell wall which is responsible Staphylococcus causing adhesion of Staphylococcus aureus with host cell and protect the cocci from the cell mediated opsonization. Now the protein A, which is the most important toxin of most Staphylococcus aureus strain, has biological property like chemotactic, antiphagocytic and anti-complement effect with the platelet damaging and hypersensitivity. So how the pathogenesis or mechanism occur? First, the protein A bind to the Fc terminal of IgG. So that what happened, the IgG FAP receptor is free. Then the protein A, which bears the Staphylococcus, mix with the IgG antiserum and there will be agglutination occur. And this agglutination is occur due to the binding of antigen and antibody. And this is alone to identify the streptococcal and gonococcal grouping. So in difference between streptococcal and gonococcal identification is done by this protein A agglutination. Then this clumping pattern also either another surface protein which present on the Staphylococcus are responsible for slide coagulants test. What is that? When the saline suspension of a Staphylococcus aureus is mixed with a slide containing 1 drop of human plasma, then the cocci of Staphylococcus are clumped. So routinely slide coagulants tests are done for the identification of a Staphylococcus aureus isolation. So the coagulants test is the standard criteria for Staphylococcus aureus isolation. Then as the bound form of a coagulants is with clumping pattern are also present in Staph aureus, so it can causes fibrinogen to fibrin. So it does not require any coagulating material as the bound form of a coagulants is present, so that there will be no other calcium or any factor is not necessary for the conversion of a fibrinogen to fibrin. Lipase also present in the Staphylococcus, which is mostly causing a number of lipid hydrolysis and causes infection of skin and subcutaneous tissue. So hyaluronidase, which is present inside the staph, it breaks the connective tissue and causes staphylokinase fatty acid modifying enzyme secretion. And also there is a protease is present, protease which helps in the breakdown of a protein, which helps in the initiation and the spread of infection. Also the nucleus, which is the heat stable and causes nucleus degradation of the cell. Then protein receptor, which bind to the IgG and fibrinogen and facilitate the adhesion of the host cell to the tissue. So by this protein receptor, the Staphylococcus enter to the tissue of the host cell. Discuss the pathogenicity of a cellular enzymes. Now we go towards the toxin of the Staphylococcus, how they causes pathogen. There is 3 types of a toxin, that is cytolytic toxin, leukocidin and enterotoxin. Cytolytic toxin is divided into alpha, beta, gamma, delta and leukocidin. Alpha himulation is the important. What it causes? It causes the lysis of RBC, mainly the rabbit RBC, not human RBC. So it is having the leukocidal cytotoxic with the dormonecrotic and neurotoxic property. So it causes the toxic to the macrophage, lysosome, muscle, renal cortex, circulatory system. And this toxin is inactivated in the 70 degrees centigrade and reactivated at 100 degrees centigrade. Now beta hemolysin. Beta hemolysin is what? It is a sphingomyelinase. It is having the cold and hot phenomena. What it means that the hemolysis initiated at 37 degree and which occur after the chilling of the body. Then gamma hemolysin, which contain 2 separate proteins and causing hemolysis. Delta hemolysin, it is action is like a detergent which effect on the cell membrane of the erythrocyte, leukocyte, macrophage and platelet. Leukocidin, which causes the membrane active toxin and causes the gamma, also contain the gamma toxin. So it is having both gamma toxin action also present in the leukocidin. Now the enterotoxin. Enterotoxin means the toxin which is intestinal. It mainly causes the food poisoning. Food poisoning symptom, that is nausea, vomiting, diarrhea. Diarrhea occur after 2 to 6 hours of eating the contaminated food containing Staphylococcus aureus. It causes the food poisoning. As Staphylococcus aureus grows in carbohydrate protein food which secrete the toxin, this toxin is heat stable and illness is usually occur by this and recovery also occur in a day. So there is no medicine required as recovery is occur within our immunity 8 types of enterotoxin is present, but the type A enterotoxin which is responsible mainly for the food poisoning, and it is identified the ELISA and latex agglutination. So in some cases it is found that post antibiotic diarrhea also found due to this. If we give any antibiotic medicines for the protection of Staphylococcus aureus infection, there is also diarrhea will find, and this diarrhea is occurred due to the enterotoxin forming by this Staphylococci. Now one important syndrome which is found in the Staphylococcus, that is toxic syndrome toxin, that is it is potentially fatal. It causes death due to the multi-system disease presenting power of this syndrome toxin. So where there is the sign, that is fever, high blood pressure, myalgia, vomiting, diarrhea, erythematous rash also found. This occur due to the group 1 Staphylococcus. The Staphylococcus enterotoxin and TSS1 are super antigen when they causes potent activation of a T lymphocyte in large scale by mitogen. So what happened, there is excessive and dysregulated immune by release of a cytokine 1 and 2, which and with interferon, gamma interferon, TNF, which causes multi-system involvement, and food poisoning with multi-system disorder will be found. One last is the 4S, that is Staphylococcus scald skin syndrome, which is due to the exfoliate skin disease due to the which occur in the outer layer of the epidermis and causes separation of underlying tissue. Mainly in the severe cases it is called as a retail disease. Okay, so this is occurs due to the exfoliating. So with this we complete our Staphylococcus aureus 20 mark question.